Hello friends, welcome back to my YouTube channel. Myself Chetan Chakradari. If you have aspirations of net zoology students, so you click the right video. So today we will cover 20 MCQs. So let's start No.
The first stable product of glycolysis is A. Glucose 6-phosphate, B. Fructose 1,6-bisphosphate, C. Glyceraldehyde 3-phosphate, D. Pyruvate. Answer C. Glyceraldehyde 3-phosphate. Explanation. In glycolysis, glucose is converted to fructose 1,6-bisphosphate and then split into two 3-carbon molecules. Glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate. Two. The end product of glycolysis under aerobic conditions is A. Ethanol, B. Lactate, C. Pyruvate, D. Acetyl-CoA. Answer C. Pyruvate. Explanation. In the aerobic pathway, glycolysis ends with pyruvate, which enters mitochondria for oxidative decarboxylation to form acetyl-CoA. Free. The enzyme hexokinase catalyzes A. Conversion of glucose to glucose 6-phosphate, B. Conversion of fructose to fructose 6-phosphate, C. Conversion of pyruvate to lactate, D. Conversion of glucose to glycogen. Answer A. Conversion of glucose to glucose 6-phosphate. Explanation. Hexokinase is the first enzyme. Four. The main site of gluconeogenesis is A. Muscle, B. Liver, C. Brain, D. Adipose tissue. Answer B. Liver. Explanation. Gluconeogenesis occurs mainly in the liver and, to some extent, in the kidney to maintain blood glucose during fasting. Five. The TCA cycle occurs in A. Cytoplasm, B. Nucleus, C. Mitochondrial matrix, D. Endoplasmic reticulum. Answer C. Mitochondrial matrix. Explanation. The tricarboxylic acid TCA cycle, also known as the Krebs cycle, takes place in the mitochondrial matrix where acetyl-CoA is oxidized to CO2. Six. The main product of beta oxidation of fatty acids is A. Acetyl-CoA, B. Malonyl-CoA, C. Pyruvate, D. Glycerol. Answer A. Acetyl-CoA. Explanation of beta oxidation of fatty acids in mitochondria produces acetyl-CoA, NADH, and FADH2, used for ATP generation. Seven. Which enzyme catalyzes the rate-limiting step of glycolysis? A. Hexokinase, B. Phosphofructokinase 1 PFK1, C. Pyruvate kinase, D. Aldolase. Answer B. Phosphofructokinase 1 PFK1. Explanation. PFK1 controls the irreversible phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate and is highly regulated by ATP and ANP. 8. The process of converting non-carbohydrate molecules into glucose is known as A. Glycolysis, B. Glycogenolysis, C. Gluconeogenesis, D. Glycogenesis. Answer C. Gluconeogenesis. Explanation. Gluconeogenesis synthesizes glucose from amino acids, lactate, or glycerol, especially during fasting or starvation. Nine. The urea cycle occurs in A. Cytoplasm only, B. Mitochondria only, C. Both mitochondria and cytoplasm, D. Lysosome. Answer C. Both mitochondria and cytoplasm. Explanation. Urea cycle begins in mitochondria, carbamoyl phosphate formation, and completes in the cytoplasm, converting toxic ammonia to urea. 10. The main energy currency molecule in cells is A. GTP, B. ATP, C. NADH, D. FADH2. Answer: B. ATP. Explanation: ATP, adenosine triphosphate, provides the universal energy currency for all biochemical reactions. (11) Oxidative phosphorylation occurs in A. Cytoplasm, B. Mitochondrial inner membrane, C. Endoplasmic reticulum, D. Nucleus. Answer: B. Mitochondrial inner membrane. Explanation: the electron transport chain ETC and ATP synthase complexes are embedded in the inner mitochondrial membrane. (12) The major site of fatty acid synthesis is A. Mitochondria, B. Cytosol, C. Nucleus, D. Endoplasmic reticulum. Answer: B. Cytosol. Explanation: fatty acid synthesis occurs in the cytosol, while fatty acid oxidation occurs in mitochondria, an example of compartmental regulation. (13) The coenzyme required for transamination reactions is A. NAD+, B. FAD, C. Pyridoxal phosphate, vitamin B6, D. Thiamine pyrophosphate. Answer: C. Pyridoxal phosphate, vitamin B6. Explanation: vitamin B6, PLP, acts as a coenzyme in transamination and decarboxylation of amino acids. (14) Which vitamin deficiency causes scurvy? A. Vitamin A, B. Vitamin B12, C. Vitamin C, D. Vitamin D. Answer: C. Vitamin C. Explanation: vitamin C, ascorbic acid, is essential for collagen synthesis. Its deficiency leads to scurvy, bleeding gums, weak connective tissue. (15) The rate-limiting enzyme of cholesterol biosynthesis is A. HMG-CoA reductase, B. Citrate synthase, C. Acetyl-CoA carboxylase, D. ATP synthase. Answer: A. HMG-CoA reductase. Explanation: HMG-CoA reductase converts HMG-CoA to mevalonate, a key regulatory step inhibited by statin drugs. (16) Ketone bodies are formed in A. Brain, B. Liver, C. Adipose tissue, D. Muscle. Answer: B. Liver. Explanation: ketone bodies, acetoacetate, beta-hydroxybutyrate, are formed in the liver during prolonged fasting or diabetes from acetyl-CoA. (17) The Cori cycle connects A. Liver and brain, B. Liver and muscle, C. Muscle and kidney, adipose and brain. Answer: B. Liver and muscle. Explanation: in the Cori cycle, lactate produced in muscle is transported to the liver, converted to glucose, and sent back to muscle. (18) The pentose phosphate pathway produces A. ATP only, B. NADPH and ribose 5-phosphate, C. FADH2, D. Pyruvate. Answer: B. NADPH and ribose 5-phosphate. Explanation: HMP, pentose phosphate pathway, generates NADPH for fatty acid synthesis and ribose 5-phosphate for nucleotide synthesis. (19) The main coenzyme in oxidative deamination is A. NAD+, B. FAD, C. CoA, D. Biotin. Answer: A. NAD+. Explanation. NAD+ accepts electrons during oxidative deamination, e.g., glutamate, to alpha-ketoglutarate. And 20. Which vitamin acts as a coenzyme for carboxylation reactions? A. Biotin, vitamin B7. B. Niacin, vitamin B3. C. Riboflavin, vitamin B2. Thiamine, vitamin B1. Answer A. Biotin, vitamin B7. Explanation. Biotin functions as a CO2 carrier in carboxylation reactions, such as acetyl-CoA followed by malonyl-CoA in fatty acid synthesis.